Well, good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Progressing Beyond Exploratory Endpoints, the Value of Digital Biomarkers in Neuroscience Clinical Studies. My name is Soumya Shashikumar and I'll be your X Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box. And we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel and that can be found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, I would also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I would like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life science companies. They offer comprehensive evidence generation solutions that combine medical imaging, ECOA, precision motion, cardiac, and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 30,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 70% of all FDA drug approvals since 2012. And now I would like to introduce our speakers for today's event. Our very first speaker here is Dr. Douglas Arnold. Dr. Douglas Arnold is a professor, and hi there, Dr. Douglas, is a professor and distinguished James McGill Professor at the McGill University, where he has led the MRI lab at the McConnell Brain Imaging Center since 1985. A pioneer in MS research and neuroimaging, Dr. Arnold founded NeuroRx in 2002 to apply advanced imaging science to global clinical trials. He continues to serve as CEO of NeuroRx, now part of Clario, where he is also senior advisor. Dr. Arnold has authored over 450 peer-reviewed publications and ranks among the top 1% of most cited researchers worldwide. Next, I would like to introduce Dr. Mark Opler, who leads research in psychiatric measurement. Hi, Dr. Opler. Psychometric validation and neuroscience at Clario. He is the executive director of the PANS Institute and co-developer of tools including SNAPSy, CGITS, and the PANS manual. His research has been supported by the NIMH, the Stanley Medical Research Institute, Brain and Behavior Foundation, and the Qatar National Research Fund. He holds a PhD and MPH in psychiatric epidemiology and completed postdoctoral training in psychiatric research. And finally, I would like to introduce Dr. Kristen Sowalski, who leads Scientific Advisory, Data Science, and Biostatistics. Hi, Dr. Sowalski, for Digital Motion Endpoints at Clario. She brings 18 years of experience in industry, academia, and clinical care, with the focus on advancing meaningful digital mobility outcomes. She holds a PhD in applied neuromechanics and a doctor of chiropractic. Dr. Sowalski also serves on the University of Florida Industry Board to support collaborative research in therapeutic innovation. Now, before I hand things over to our speakers, I would like to present the agenda for today. First, Dr. Arnold will dive into improving imaging endpoints, examining how advanced brain imaging is shaping regulatory-ready measures for multiple sclerosis and other conditions. Next, Dr. Sowalski will talk about enhanced movement assessments, learning how wearables and composite scores provide consistent, reliable measures in movement disorder trials. Dr. Opler will then discuss advanced psychiatric studies and how vocal biomarkers provide scalable objective measures for depression and schizophrenia. Finally, we'll wrap up and open it up for questions and answers. And so, without further ado, I would like to hand the mic over to our first speaker, Dr. Douglas Arnold. You may begin when ready.
Hi, everybody. I'm very pleased to be able to speak with you today to share some of the exciting new developments in imaging biomarkers for drug development in MS. Have the next slide, please. What's emerged in recent years with respect to MS is an appreciation for the fact that there are two relatively independent biologies that underlie the symptomatic evolution of this disease. And both biologies are present in essentially every patient, but to very different degrees, particularly at different times or different durations of the disease. This has actually important implications for drug development, which I'll explain in a few minutes. The next one, please. Most patients begin with a relapsing remitting course of MS, and this is due to focal inflammation in the white matter, primarily due to the infiltration of peripheral inflammatory cells. And in the right-hand panel, you can see a blood vessel with inflammatory cells invading the brain surrounding that blood vessel. These are peripheral immune cells. They're coming into the brain from outside. On the left, you can see an MRI that corresponds with this kind of pathology, and there are multiple white spots in the brain corresponding to these focal areas of inflammation. You can think of them like a pimple, really. They're focal, highly inflamed, and then they get better and go away, but they can leave a scar behind. The next one, please. There has been a tremendous success in treating this aspect of MS pathology. And what this slide shows is the effect of an anti-CD20 monoclonal antibody on lesion development in patients with relapsing remitting MS. And in the darker bars, you see the number of lesions during the first 6 months, 1 year, and then 2 years of a clinical trial. And in the lighter pink colored bars at the bottom, you can see the effect of the drug in suppressing these lesions. And the suppression is almost complete with this therapy. And in fact, it's almost complete with a number of other therapies related to monoclonal antibodies, either affecting the same target, the CD20 B cells, or other aspects of infiltration of peripheral immune cells into the central nervous system, like transport through the blood-brain barrier. The next one, please. The problem that remains unsolved is shown in this slide, which is a Kaplan-Meier curve that shows the progression of disability, the increase of disability in patients on placebo in gray and on the same therapy I just showed you in blue. And despite the almost complete suppression of new lesion formation, these patients continue to develop clinical disability. The next one, please. So the suppression of focal lesions is clearly not the answer to preventing progressive disease, and we need to understand the basis for that pathology, and then we need new targets for treatment in order to suppress that pathology. The next one, please. One of the main candidates for this kind of pathology is ongoing focal inflammation in the brain despite the suppression of new focal inflammation. So here on the left, you see the suppression of new lesion formation, which is down by 99.2% 2 years after starting therapy with ocrelizumab or anti-CD20 monoclonal antibody therapy. And on the right, you see the total non-enhancing T1 lesion volume. Now, T1 it's an MRI contrast that's associated with a relatively severe tissue destruction. And you can see that the volume of tissue being destroyed within lesions is increasing progressively in the placebo-treated group in gray. That's not surprising, but what is surprising is that it continues to increase in the patients treated with the anti-CD20 therapy, despite the fact that there are no new lesions forming. Could I have the next one, please? Now, how can this happen? What this slide shows is the histology or the histopathology of what are called chronic active lesions. So these are lesions which are no longer actively inflamed in their center, but they have a rim of activated microglial cells, which are resident inflammatory cells in the central nervous system. And these cells, which are different from B cells and don't respond to the anti-CD20 therapies, continue to break down myelin, which you can see in the right-hand panel, and continue to damage and destroy axons at the edge of these lesions. The inflammation is not completely restricted to these kind of lesions, but it's most evident in these kind of lesions. The next one, please. This is an MRI video loop showing the effects of such pathology on the MRI. And in the left side, we see a T2 weighted image, and on the right side, a T1 weighted image. And if you look at the right side in the T1 weighted image, you can see, and we're looping through this, these scans at 6 months, 12 months, and 2 years. You can see that the lesion is getting progressively bigger, but in a relatively subtle way. The next one, please. Because we understood or we wanted to understand the development of this kind of pathology in order to understand why the T1 lesion volume was continuing to increase in patients who had no new lesion formation, we developed at NeurRx a new imaging biomarker of what we call slowly expanding lesions. Okay, and the way this is done is to register or overlap two images at different points in time. You see them on the left two panels, baseline and week 96. And then calculate the deformation field, the distortion that has to be applied to every voxel of that image in order to make the second image match the first image. And then you can use fancy mathematics, shown in the colored image at the right, to calculate what's called the determinant of the Jacobian, which is a direct measure of the volume change in each voxel of the image. And from this image on the right, we can detect lesions that are undergoing slow expansion, concentric expansion over time, that are not acute. Acute lesions would get bigger and then get smaller. These are lesions that are getting progressively bigger in a concentric fashion. The next one, please. The slowly expanding lesions are one marker of the kind of biology that is associated with progression. Another marker is called phase rim lesions, or pearls. Okay, and this, unlike these slowly expanding lesions, it requires a special MRI acquisition in order to detect or to be sensitive to subtle changes in phase at the circumference of these lesions. And this is called susceptibility weighted imaging. It measures magnetic susceptibility, or the magnetic field gradients, really, at the edge of these lesions. The next one, please. And with with specialized sequences like this, it's possible to identify lesions such as you see in the left-hand panel with the dark circle around it, and this is highlighted in the right-hand panel in green, where the phase rim is shown in blue surrounding the green. So if you look back now at the left-hand image, that circle of dark rim around the lesion is due to an increased distortion of the magnetic field surrounding that lesion. And this is due to the accumulation of iron in the inflammatory cells at the edge of those chronic inflammatory lesions. Now, they don't all have iron, but about half of them have iron. And that half of these lesions can be visualized using this kind of susceptibility weighted imaging. The next one, please. So what I've tried to explain to you is the reason why most of the trials that are coming in MS will focus on progressive disease. I mean, there are some trials looking at generics, for example, for relapsing remitting disease, but the action in MS drug development is focused on progression, where new targets and new molecules are required. And in order to efficiently determine whether those drugs are doing what they're supposed to do, we need biomarkers of progressive biology that will support the biologic efficacy of those new treatments in clinical trials, phase 2 and phase 3 trials. And there are a lot of such trials that are ongoing at the moment or just starting, because there's a lot of interest in developing new therapies for this aspect of the disease. At Neuroscience, we're doing multiple trials of Bruton's tyrosine kinase inhibitors, excuse me, of CAR T cells where a cell therapy is being applied to overcome one of the limitations of the monoclonal antibodies, which is penetration across the blood-brain barrier into the brain. And there are other approaches as well, anti-CD40 ligand therapies, small molecules directed against TREM2 or TYK2, vaccination against Epstein-Barr virus, which is thought to be associated with MS and potentially causal, and other treatments as well. In all of these cases, it's extremely useful to have a biomarker that is more sensitive than the clinical outcomes that will show that the drug is doing what you want it to do, which is suppress this chronic inflammation that is compartmentalized in the brain and not due to the infiltration of peripheral inflammatory cells. So I hope that you've got a somewhat of an appreciation now for the evolution of novel biomarkers in this field and their usefulness in MS drug development. Thanks very much.
Thanks, Dr. Arnold. Well, I'm going to speak a little bit about movement analysis, particularly using wearable sensors to capture endpoints of movement. It's very important to relate the pathology that we see in MS through imaging to behavior. So I'll speak really particularly around motor behavior or movement performance, how well someone moves around in their environment, which affects their quality of life. Next slide, please. So the way movement has traditionally been captured in clinical trials has been through conventional scales. So these scales are still important, such as the EDSS, right? We definitely want to capture the clinical impression as well as the patient perception of what's going on. So, but in addition to these items, we want to be able to actually measure movement directly. So we want that clinical impression, the patient impression, and then we want the ability to measure movement in ways that we can't perceive with the human eye. Next slide, please. So one way that this can be done is by applying wearable sensors and having a person perform simple tasks like a 2-minute walk test or a 30-second stand test or a quantified sit to stand test or tug. So by putting a multi-sensor array on various parts of the body, you get a wide variety of endpoints that are available. And in doing so, you can get very specific endpoints to particular indications. So we apply this in different indications. So if we go to the next slide, examples of this include Parkinson's disease, right? This is a hypokinetic movement disorder, meaning movements become smaller and slower. So we see sort of a signature movement with Parkinson's disease, and that's reflected by what the wearable sensors measure. We see disruption in balance. We see reduced arm swing. We see a slower turn velocity relating to that bradykinesia. That's that primary a motor symptom in Parkinson's disease. And then we see smaller movements by decreased stride length. But if we look at a different indication, such as ataxia, these endpoints are going to look different. In ataxia, there's disruption of coordination, right? So we see kind of larger, more variable movements, and we see an increase in sway amplitude. We see increased time spent in double support, meaning 2 feet on the ground, so they're trying to maintain more stability. Overall, increased variability because of that impairment of balance while they're walking. And then we see that step height, that kind of staggered gait. And then going back to the example of multiple sclerosis, there's a disruption of the signal, right, from the brain to the body to move. And so we see different endpoints here representing multiple sclerosis, looking at a sway that's impaired or balance, but in different directions than the other two. We see movement of the trunk going side to side, and we see disruption in turn measures, even with normal gait speed. So if we go to the next slide, we want to be able to relate, again, these measures with imaging to movement measures. And there's been some work done in this area. This first study showed that poor balance control is actually related to reduced white matter, microstructural integrity of cortical proprioceptive tracts in multiple sclerosis. So in this instance, a wearable sensor was placed across the lower back, and 2 balance tests were performed, a 30 seconds eyes open task and a 30 seconds eyes closed task, to look at the ratio and the Rauburg quotient. And what was found that the poor proprioception through this test was related to that poor tract microstructure. And then there's another study that's been demonstrating in MS. Oops, one more. Thank you. That cortical thickness and inhibitory activity are also associated with reduced turn performance. So turns can be quantified with wearable sensors by putting a sensor on the lower back as well, and sensors on the feet to look at steps in a turn. And so turning impairment was related to these structures as well. Next slide, please. So while we're capturing movement endpoints, we want to make sure they actually matter to the patient, right? We need to understand the pathology, we need to understand the behavior, but does this matter to the patient? Well, one of the #1 things that does matter to everyone is falling, right? We know that falls is linked to further injury, further hospitalization, increased healthcare costs. So we want to understand the relationship of these measures to falls. And what we saw here in this study of 86 people with MS who completed fall surveys, they did a classic 25-foot timed walk test and then movement analysis with sensors. And what was determined that the falls were associated with worse balance metrics, including sway area. So how much somebody, their range of motion is moving around sort of forward to back and side to side while trying to maintain their balance. Jerk, meaning the disruption of the movement. When you stand still, there's this nice smooth sway pattern. It becomes more jerky in nature and disruptive in certain indications, including MS. So these measures were related to falls, in addition to the slower 25-foot walk test. But if we look even earlier, before there's the disruption of a slower walk test, we can see some of these other endpoints that demonstrate impairment earlier. So if we go to the next slide, this is another study that was performed in which the timed 20-foot walk test did not demonstrate difference between controls and subjects with MS as far as the time it took to walk. But at the same time, we did see differences in some of the balance measures and the walking measures. So on the left side of the screen, you'll see a typical 30-second sway test in which a sensor is worn on the lower back. So in the bottom, you see what's called a stabilogram. In a control subject, you have a little bit of forward and backward movement and some side-to-side movement, and that path is fairly smooth. Now, in the MS subjects, we see this area increase because they have difficulty with balancing. So they're tipping forward and back more and side to side more. The trace is a little bit more jerky in nature relative to the control. So we see this even when we're seeing a walk test as quote normal as far as speed goes. In the walk test, what we also see, if someone walks 7 meters in turns back and forth for 2 minutes, those turn durations were decreasing, and that lateral trunk range of motion was actually increasing. So while we some of the traditional scales that have been performed are testing as normal, we want to start wearing sensors so that we can detect signals earlier. That way treatments can be targeted earlier as well. In the next slide. I'm going to move on to another example, another indication. MS has quite a bit of research done with wearable sensors, but probably the most that's been done to date is in Parkinson's disease. So we know quite a bit more in this area. And out of all the hundreds of measures that you can collect by wearing multiple sensors on the body, it's been revealed that there's a top subset that really defines Parkinson's disease, that discriminates it from healthy controls. So some of these top measures are that slower turn velocity, that reduced arm swing range of motion, and then decrease in the foot strike angle. We know a classic symptom of Parkinson's disease is that shuffling, so that's a good measure of that, and that can be detected earlier than the human eye if you have a sensor that is objective and very reliable from test and test retest. What's interesting and what we want to see is that these measures also relate to some of the conventional scales, right? We want that to showing similar progression. We don't want a perfect relationship because then it would not be additive. By adding the sensors, there's more wealth of information available. But we do see these three measures relate to the clinical scale. So the MDS UPRS total, as well as the motor part 3, we see in the middle section on the right, a little bit small, but relating these measures to turn velocity, and then also relating them to the patient perception, right? The MDS UPRS part 2, which is interviewing the patient how they feel about these symptoms, as well as the PDQ-39 more quality of life questionnaires. So these measures are relating to things that the clinician is seeing, as well as how the patients are feeling. But it's adding value by quantifying things earlier than sometimes perception can detect. So in the next slide, this is just more specific to that 30-second balance test in Parkinson's disease, right? Though that previous slide was more around the gait, the 2-minute walk task. But similar to MS, we see a changed pattern in PD very early on compared to controls. So we see that movement really being increased overall total sway area, and that measure of jerk, the jerkiness of that pattern there is also increased relative to controls in Parkinson's disease. In the lower left-hand figure, this is an untreated PD subject, and this demonstrates how that progression of the balance impairment occurs over time in just 1 year. You can see that visually, that sway area is increasing over that time. And it's been tested whether eyes open or eyes closed is the best discriminator in Parkinson's disease. And in this case, eyes open is showing that greater difference between controls and Parkinson's disease. And then in the next slide, we will see that both of these types of tests, a simple 2-minute walk test and a 30-second sway test, can be used in parallel with machine learning to track disease progression in Parkinson's disease earlier than the conventional scale, the MDS UPDRS, right? Again, this is not to replace, this is to be additive to that clinical impression and that patient perception of their experience with their movement disruption. But what's great here is by adding those sensors, we detect progression at 15 months and at 18 months when the conventional scale does not. So that means that's a great opportunity to test therapeutic efficacy of interventions that are targeting progression in Parkinson's disease. And then in the next slide, you'll see those same 2 tests have been used as well. These studies are coming out of the University of Oxford. It's from the OXPIP study data with Dr. Kristalina Antonades and her great team. Those same 2 tests have been used to predict future fallers up to 5 years in advance. So we're seeing a lot of value by adding these in studies and clinical trials to both detect things earlier and then prevent things that are really meaningful to patients. So if we go to the next slide, there's another natural history study done called the WATCH-PD study. And so in this study, there were tasks performed in the clinic as well as remotely. The in-clinic tasks with sensors on multiple locations in the body also revealed that a 2-minute walk test shows progression over 12 months, in particular components of gait, that stride length, overall gait speed, and arm swing. So we're seeing the repeated same measures rise to the top that we really want to track when we're looking and evaluating treatments. In addition to that, which has a little bit, there's a lot of evidence in gait, there's newer evidence around bradykinesia, tremor, things of that nature. But when we looked at the digital score of bradykinesia with 2 tasks, that pronation supination task and toe tapping, and a composite score combined with that, that also demonstrated significant progression at 12 months in PDs versus controls when the MDS UPRIDs did not detect that. So just we're accumulating more and more evidence to add these to these studies. Okay, next slide. All right, and then another fantastic study came out, another one with a composite digital measure, right? The MDS UPRIDs is a giant composite measure, but we know it's subjective in nature, right? It's the clinician, it's the patient, and so there's gonna be some natural inherent variability in that. But by creating a composite score with the data that comes off of these wearable sensors, what we see is a similar, in this case, a similar track of progression, but reduced variability. And so the effect size for the digital composite score was 2.14, whereas the MDS UPRIDs Part 3 was 1.41. So what that implies is that less subjects are needed in a study to power that and see these measures of progression. So overall, by adding the sensors, we're seeing earlier detection of progression, so we can target these things earlier to provide better treatments for patients, and then a reduction in the number of patients needed to get that signal. So shorter duration of trials, smaller trials, to provide what the ultimate end goal is, providing effective therapies to patients sooner. And in the next slide, there are also ways available now to measure movement out in the real world. Everything that I spoke of previously was in a controlled setting with multiple sensors on to get a very defined sort of movement signature, but we also need to understand how this relates out in the real world, right? So in this, we want to balance using the right number of sensors for compliance out in the real world versus precision. And a wonderful consortium, the Mobilized D Consortium, has done a wonderful amount of work with verification and validation and different cohorts, providing endpoints of gait and turning out in the real world, just with one sensor on the lower back. So it's important to have both of these modalities in future clinical trials. And with that, that concludes my portion, and Dr. Opler will continue on speaking a little bit more about adding more than some of the subjective conventional scales to help assist these trials.
Thank you so much, Dr. Sawalski. Much appreciated. So I'm going to be speaking a bit today about the use of vocal biomarkers in psychiatry trials. If we go to the next slide. So, one thing I want to reflect on for just a second, my colleagues have been kind enough to talk about neurologic disorders like multiple sclerosis and Parkinson's disease. When we get into psychiatry, we are, even before we get into the digital world, we're saddled with some serious issues in measurement. We're trying to measure often very subjective phenomenon. Many of the existing tools that we use in psychiatry studies are somewhat old, somewhat aged, developed in prior eras when treatment and also perspectives on disease were quite different. The tools that we typically use, especially as primary endpoints, are often difficult to administer. They usually require clinical interviews lasting anywhere from 20 minutes to an hour plus. So in addition to being subjective, they're difficult to apply in a fast-moving study. or in many of the models that the industry wants to evolve into, such as distributed or digital health or decentralized trials. So to put it bluntly, our measures are also generally, in addition to being subjective, they're noisy and they're prone to failure. They are often difficult to use. They require a very high degree of training and a lot of calibration. So the response, I think, the natural response of almost everybody involved in these studies from time to time is surely we can come up with something better than this. Needless to say, the idea of turning to digital tools, wearables, apps, or AI-driven methods comes up very frequently. Let me say just a word or two about the current view and some of the hesitation around moving in that direction from an industry and I think also from a scientific perspective. Go to the next slide. So, I think it's fair to say that the application of artificial intelligence in health has a mixed record overall. The index example, which everyone here is doubtlessly very familiar with, was first illustrated in work with IBM Watson on oncology. I won't dive deep into this, but it's simply a reminder to all of us, myself included, that we have to be somewhat careful and we have to be thoughtful when we apply any new digital health technology to any of our existing measurement problems. So, in this example, to put it as simply as possible, the Watson tool was being used in oncology to try to create either prognostic or recommendation or treatment recommendations or similar solutions for patients. They were trained on a set of limited hypothetical data sets. And the results, unfortunately, as some studies and many newspapers reported, were ungeneralizable, meaning that the results were not consistent. Comparison, for example, to expert evaluation or expert recommendation, concordance was quite low in some cases, as you see here. So again, one example now somewhat dated, but it's made everybody, I think, very rightly cautious. And there have been other examples since, I think, that make us all want to make sure that we're standing on very firm ground before we move from a tried and true method to one that is more, shall we say, digital or AI driven. Can we go to the next slide, please? So there has been an awful lot of work on what I'll call digital biomarkers in psychiatry. So broadly writ, these tools cover a lot of ground. They include wearables, as discussed by Dr. Sawalski. Some of them involve imaging or variations on a theme of imaging, as discussed by Dr. Arnold. There are a lot of different tools. There's been a lot of very interesting research, and unfortunately, to date, the impact of all this development on our clinical trials ecosystem has been limited. Few, if any, of these tools have moved beyond exploratory, and for various reasons, they're not being widely adopted. There's a nice quote from a paper back in 2018 that I've cited here that I think still merits consideration, and it quite simply says the field's full of enthusiasm, but we need more data and rigorously designed studies to displace the hype. So, in the work that we've done at Clario, this has been our North Star. This has been our guiding principle in psychiatry. And I'm going to talk to you a little bit today about how we're trying to move beyond exploratory and move from hype to something more meaningful. Can we go to the next slide? So in our development, we asked ourselves, what's the matter with biomarkers, and especially digital biomarkers in psychiatry? Why aren't they more widely used, and why aren't they more central to the work we're doing? And after a lot of thought and talking to colleagues on the sponsor side, on the CRO side, at research sites, we came up with sort of three big challenges that have prevented these measures from making their way into our studies in a more thorough fashion. The first is implementation. Wearables and other devices, I think, certainly have their place, but many research sites and many patients and caregivers often find them challenging to implement. whether they're wearables or apps. Perhaps this has to do with the populations that we work with in psychiatry. Perhaps it has to do with the way that they've been developed. Hard to say, but it has been identified as a challenge. The second is that these measures, from the perspective of a therapeutic area that already works in very subjective and often very noisy tools, they're generally considered somewhat complex and hard to analyze. In some cases, it wasn't clear what their relationship was to the disease, and it wasn't clear what their relationship was to stakeholders, including patients and caregivers. So that complexity and that lack of transparency also made it difficult to make a case for them. The third piece, and I think this is one that everybody will appreciate, is that the purpose of these things and their application in our studies was not well defined. The reasons for doing so, beyond a general desire to move beyond traditional subjective measures and the problems that they represent, beyond that general objective, it wasn't always clear in our early experience with digital biomarkers what they were for. So, with that in mind, we started asking the question, what would the perfect digital biomarkers battery or the perfect digital biomarker itself look like in psychiatry? Let's go to the next slide to take a look. What we heard in our conversations and what we arrived at ourselves was that, a good digital biomarker should be meaningful, it should be useful, it should solve a real problem, and for preference, it should be, so simple as to be almost invisible. So we need things, again, just to recap, we need tools that are not complicated to implement when we're dealing, especially with a large study of patients experiencing behavioral health issues. We want to have relatively easy, an easy path to analysis. We want to understand what it is we're analyzing. We would like, if possible, to be analyzing more objective, less noisy data. And finally, we want the tools that we implement to have a very strong relationship to things that we care about, especially our scientific objectives. So with that in mind, let me present a little data on what I see as, probably maybe not the best, but certainly the first path forward for the evolution of these tools. If we go to the next slide. So I'm going to talk a little bit about speech latency. So this is the speed, rate of production. There are lots of different kinds of speech latencies, including things like time to speak, reaction time. They're related to lots of different pathologies in psychiatry, especially depression. And there are lots of different ways to look at it. It can be extracted from recordings of free speech, where people are just recording, say, an audio diary, structured interviews, unstructured conversations. We can extract this measure out of lots of different material. And again, it's been well studied, even predating the digital era. It's even older than I am, just to put it in context. Let's go to the next slide. The specific speech latency that I'll talk a little bit about is speech turns latency. So, in, let's say, a clinical interview or a conversation, it's the time between, in this case, the participants being asked a question and the point at which they react, right? So how long does it take a participant to start responding to a question after you ask them? So when we measured this space between the question and the response, and we looked at it in a large phase 2 trial of bipolar depression, we discovered something very interesting. We discovered that we had recordings of every clinical interview of the primary outcome in one of these trials, an instrument called the MADRS. And what we found was, in this relatively large 7-country study, in every single country, when we calculated the speech turns latency of the patients, those who were depressed uniformly had longer speech latencies, longer times to response than the patients who weren't depressed. When we tried to model the relationship between speech turns latency and the MADRS total score, which was the primary outcome from the study, looking especially at post-baseline data after randomization, we discovered a very strong predictive relationship between these two phenomena. Let's go to the next slide. As fascinating as all this wasn't really the bolt of lightning. The thing that I think told us that something real was happening here was what happened when we actually classified patients at screening into two categories: those who had long latencies, whose latencies certainly put them in the upper range, and those who had shorter, more normal latencies. We term these vocal biomarker positive for those who had longer speech turns latency, and vocal biomarker negative for those whose speech turns latency sound less depressed and more normal. And what we found was this status actually predicted their treatment response in the study. And when we stratified them into two groups based on their VBM classification, we saw something extraordinary. The separation between drug and placebo, especially the high dose of the drug being used in the study, in this case shown in black on this graph. No, sorry, the black is placebo. The two doses of drug on the study are shown in green. Basically, what we saw was separation between drug and placebo was vastly improved in the VBM positive patients, whereas the VBM negative patients didn't really show much difference at all. It suggested that the VBM patients had more placebo response, and also that there may have been a mismatch between their reported depression while being administered the MADRS and their objective depression as measured by speech turns latency in their voice. As interesting as this was, we knew that lots of findings like this generally may not replicate. We may see it in one study in one population, may not see it in another. Perhaps our model building was bad, maybe we dabbled in overfitting, hard to say. As exciting as it was that we saw an increased effect size when we applied this, would we ever see it again? As luck would have it, we did. Let's go on to the next slide. When we applied the same general strategy in a study of schizophrenia, looking at acute schizophrenia in patients being treated with brilaroxazine in a study sponsored by Reviva Pharmaceuticals, we saw essentially a replication of our earlier finding. Those VBM positive patients exhibited remarkable response to the drug. VBM negative patients did as well, but it was much more pronounced in the VBM positive folks. So again, here, especially in the high dose of the drug shown in purple, that solid purple line on the bottom on the graph to the left, you'll see that the effect size is extraordinary and can be measured as early as day 7 in this group. So this told us that something real was going on. And when we dug in further and started looking at phenomenon beyond just acute psychosis, we saw some interesting replications, again, of the same thing. If we could go to the next slide. Once again, we saw remarkably similar results. Now, let me say just a little bit about these findings. So whereas the previous figure that I was showing you was overall symptom severity in a study of acute psychosis, this slide and these figures are focusing very specifically on negative symptoms. So these are the deficits that we often see in patients with schizophrenia, ranging from lack of interest in everyday activities to lack of facial affect, lack of emotional responsivity, all the things that make it difficult for patients to engage with the world, and often makes it difficult for them to resume the lives they had before they got sick. So this is a very important cluster of symptoms. It's very, very difficult to treat, I should say as well. There are lots of examples of well-conducted but negative or failed trials in this area. So the fact that by applying, again, similar model with an objective, relatively simple marker of speech latency, we could produce a result like this, I think it is something of an accomplishment. Now, as I said before, replication is key. While response in negative symptoms is one of the holy grails in psychiatry and clinical trials, and as exciting as the result is, we need to continue being rigorous, we need to continue replicating, and we need to continue making sure that we're doing this right. So why don't we go to the next slide? I want to say just another word or two about different applications. So what I've discussed so far has principally been about enrichment, right? So the use of, let's say, a vocal biomarker at screening or pre-randomization to classify patients objectively, either to augment or to supplement existing inclusion criteria. That's what the work we've done has suggested, and that's a strategy that we're pursuing. But there's more. We also want, as I said before, to try to improve on or overcome the limitations of our existing endpoints. So we have also been looking at sensitivity to change. In the study I mentioned before on bipolar depression, we also wanted to look at not just the classification according to type in VBM positive, negative, but also the change in speech latency over the course of time in the trial. And one thing we noticed is that, in patients whose depression remitted during the trial, when we compared the change in speech latency from the beginning of the study to the end, then in both treatment arms of the drug being used, there were some very interesting findings. While placebo showed statistically no meaningful difference between from drug, basically patients on placebo, whether they were remitted or not, their speech latency didn't change much. You'll see a little bit of change, but it's about the same in the remitted and the non-remitted patients in the placebo arm. However, in the treatment arms, the patients who responded to treatment, both on the low dose and the high dose, showed dramatic changes actually. Their speech latencies improved quite a lot. They started to become almost what I will broadly classify as almost normal. So from where I stand in a field where we've got difficult to use, subjective, noisy measures, this is a step in the right direction. We have a lot of work to do if we're going to catch up with our colleagues in neurology. We're nowhere near, I think, the quality of the data or the precision that we see in either imaging or in the precision movement work presented, but we're doing our best. So on that, I think that's all I have to say. I want to just sum up now for my key points, if I may, on the next slide. To move beyond exploratory, we've got to make digital biomarkers in psychiatry as invisible as possible, as I mentioned before. So, capturing them via existing technologies. In this case, extracting them from clinical interviews already being conducted using eCOA, and then running that in real time or near real time to evaluate will allow us to do things that we never could have imagined. Even if we're not yet ready to let go of our old, somewhat creaky measures, the opportunity for signal enhancement, enrichment, and similar strategies, I think, has arrived. So on that note, I think I'm going to stop. I think there's a wrap-up slide on the next panel. So key takeaways from today, I think from all of us here. First, we believe that we can optimize trial efficiency by using digital biomarkers strategically. Improving effect sizes in psychiatry or in neurology with these tools will reduce sample size and shorten study timelines. Next, I think it's very clear from Dr. Arnold's talk that advanced imaging is shaping our endpoints. Brain imaging techniques that support regulatory-ready endpoints in neurodegeneration in studies and beyond are here. Next, as Dr. Sawalski described, we can enhance our movement assessments like the MDS-UPDRS with wearable sensors and composite scores that deliver far more reliable measures and help us again achieve better effect sizes, small studies. And finally, as I tried to say, we can advance our psychiatry studies as well with vocal biomarkers that are objective and scalable for the moment for depression, schizophrenia, and soon maybe for more. On that note, I'll pass it back to our moderator, and I thank you all for listening.
Well, thank you very much for that very insightful presentation. Now, I would like to invite our audience to continue sending their questions or comments for the Q&A portion of the webinar. If you are attending via GoToWebinar, please use the questions window. For those of you watching on LinkedIn Live events, feel free to post your questions in the comment section. I have already received some questions, so I'll start with those. Now, the very first question that we have here, Dr. Arnold, I think this may be for you. This person is wondering what challenges do sites face when acquiring specialized MRI sequences needed for these advanced biomarkers. Dr. Arnold, I think you're muted. If you can try and unmute yourself.
Sorry, I realized that I'm
That's
unmuted.
okay.
It's a good question, but in fact, with respect to the slowly expanding lesions, there is no challenge whatsoever. These are calculated using the conventional images T1, T2 flare that we acquire in every MS trial anyway. The analysis is based completely on advanced post-processing of the images after we receive them. So there is no friction whatsoever. With the phase rim lesions, there is a minimal amount of friction. The sequences that are required, the susceptibility weighted images, are not standard sequences. They have to be implemented on the scanner at every site. But we have the experience to specify exactly what parameters need to be input in every scanner make and model so that the appropriate images can be acquired. Having said that, there is a minimal additional burden on sites because they would have to, after implementing these sequences, they would have to do a dummy run with the logistical, not challenges, but just the logistical issues associated with that. Whereas, if they were, for example, doing ongoing studies that didn't include susceptibility weighted imaging already, they might get approved for a new study without having to do a dummy run, but that's it.
Thank you so much for that answer. Now, Dr. Sowalski, someone sent this in for you. They are wondering, can the same movement assessments in the clinic be performed remotely? And if so, does this impact the quality of the data?
Yeah, that's a great question. The technology is there. You can do like multi-sensor array in the home setting. I think this push kind of came about specifically too during the COVID epidemic and the need to try to continue trials. And so I think there's some lessons learned in that. While it can be performed, you really need to assess the pros and cons, right? Naturally, when you bring this technology into the home, it puts more of that responsibility onto the patient. So in indications, say, right, we're like in psychiatry where there's some difficulty with cognition and things of that nature, it's going to make that more difficult, right? But maybe some earlier phase disease where there's not a cognitive impairment, it's more likely that that's going to be more successful. But things to consider too are space requirements, and that you don't have that quite as controlled, right? You definitely want repeatable, reliable conditions as well as the sensor. So I think it's available, but it's a conversation to be had, with the goals of the trial. Is it a symptomatic treatment where you truly need these assessments sort of a daily, or if it's progression over time, finding that balance of, is it better just to have it, say, quarterly in the clinic where it's controlled, less burdensome, or is it worth that and the population can handle it to get that data? So again, it's there. It's really worth a conversation to say what the indication is and what the goals of the study are to use it. Yeah, I think that the easier solution is probably going to be one sensor worn passively in the home to complement the in-clinic to kind of get both pictures most successfully. But again, the tech is there, and I think the data is still sort of coming out around what that looks like in the home.
Thank you so much for that. I think we have time to squeeze in one more book question. And Dr. Opler, I think this one might be for you. This audience member wants to know, can we use vocal biomarkers methods in areas other than psychiatry?
That's a great question. My theory is yes. My belief is that the same things that we're measuring with patients with depression or psychosis are probably not specific necessarily to that disease. For example, I'm quite sure that speech in Parkinson's, which is an area of a lot of development, and even in MS as well, two areas I'm definitely not qualified to speak about. It would be fascinating to study these same measures there as well and see what they can tell us. So short answer, yes, but stay tuned for updates.
Well, thank you so much for that answer and for all of the answers. We have, however, reached the end of the Q&A portion of this webinar. If we couldn't attend to your questions, the team at Baclario may follow up with you. Or if you have any more, please direct them to the email address on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our webinars. Additionally, I also have shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us once again in thanking our speakers for their time here today. We hope you found this webinar informative. Have a great day, everyone, and thank you for coming.
Bye, everyone.